Critical thinking questions on plant cell. 1. Which element of the cell theory has practical applications in healthcare because it promotes the use of sterilization and disinfection? A. All cells arise from pre-existing cells. B. All living organisms are composed of one or more cells. C. A cell is the basic unit of life. D. A nucleus and organelles are found in prokaryotic cells. 2. What are the advantages and disadvantages of light microscopes? What are the advantages and disadvantages of electron microscopes? 3. Mitochondria are observed in plant cells that contain chloroplasts. Why do you find mitochondria in photosynthetic tissue? 4. In what situation would the use of a light microscope be ideal? Why? 5. The major role of the cell wall in bacteria is protecting the cell against changes in osmotic pressure, pressure caused by different solute concentrations in the environment. Bacterial cells swell, but do not burst, in low solute concentrations. What happens to bacterial cells if a compound that interferes with the synthesis of the cell wall is added to an environment with low solute concentrations? 6. What determines the upper limit of cell size? 7. Which of these is a possible explanation for the presence of a rigid cell wall in plants? A. Plants remain exposed to changes in temperature and thus require rigid cell walls to protect themselves. B. Plants are subjected to osmotic pressure and a cell wall helps them against bursting or shrinking. C. Plant cells have a rigid cell wall to protect themselves from grazing animals. D. Plant cells have a rigid cell wall to prevent the influx of waste material. 8. Bacteria do not have organelles, yet, the same reactions that take place on the mitochondria inner membrane, the phosphorylation of ADP to ADP, and chloroplasts, photosynthesis, take place in bacteria. Where do these reactions take place? A. These reactions take place in the nucleoid of the bacteria. B. These reactions occur in the cytoplasm present in the bacteria. C. These reactions occur on the plasma membrane of bacteria. D. These reactions take place in the mesosomes. 9. What are the structural and functional similarities and differences between mitochondria and chloroplast? 10. Is the nuclear membrane part of the endomembrane system? Why or why not? 11. What happens to the proteins that are synthesized on free ribosomes in the cytoplasm? Do they go through the Golgi apparatus? A. These proteins move through the Golgi apparatus and enter in the nucleus. B. These proteins go through the Golgi apparatus and remain in the cytosol. C. The proteins do not go through the Golgi apparatus and move into the nucleus for processing. D. The proteins do not go through the Golgi apparatus and remain free in the cytosol. 12. Inhibitors of microtubule assembly, vinblastine for example, are used for cancer chemotherapy. How does an inhibitor of microtubule assembly affect cancerous cells? A. The inhibitors restrict the separation of chromosomes, thereby stopping cell division. B. The inhibition of microtubules interferes with the synthesis of proteins. C. The inhibitors bind the microtubule to the nuclear membrane, stopping cell division. D. The inhibitor interferes with energy production. 13. How does the structure of a plasmodesma differ from that of a gap junction?